This is the first lecture for my Bio 412 Comparative Genomics course. And this is just going to be sort of a general introduction to a lot of the sort of conceptual frameworks that we're going to be using throughout the rest of the class. It's a little bit of a long lecture, so I may end up breaking it up into pieces, but we'll see how it goes. So for this, hopefully after this lecture, students will have a good understanding of what comparative genomics is, a good understanding of the basics of molecular biology and genetics and how it relates to this subject matter of genomics, an appreciation for the role that the various components, especially repetitive elements, can play in a genome and species biology. So an understanding that it's not just genes that are what's important in the genome. And then finally, have a good understanding of some of the other related fields that we associate and apply in the field of genomics, things like proteomics, transcriptomics, epigenetics, things of this sort that we'll also cover in this class. So first I just want to remind the class that I'll be following along in the textbook for the course, which is Introduction to Genomics, 3rd edition by Andrew Lesk. It's a good reference, and I hope that it will serve you well. So if you need some context, a lot of the lectures will deal directly with a lot of the content that you might also find in this textbook. So we're in the era of genomics, right? All you have to look at the thousands and tens of thousands of COVID genomes that are sequenced almost every single day in the world right now. Technological advancement has led to a massive decrease in the cost of sequencing. And if we look at it over time, and we think about what it costs to sequence a megabase or 1 million bases of DNA, we can see that cost has pretty much been falling exponentially for a pretty good period of time. It's kind of leveled off now, but it's just a matter of time before there's some sort of new innovation and it pushes it even farther. These days, it's not so much that the cost can even go down that much farther, given how low it is. It's more that what we're looking at is ways for us to scale up the amount of throughput that we're able to put through. And a lot of the costs right now that we face is actually more how do we actually process this data, not how do we generate it. So because we've had this huge decrease in the cost of sequencing, it has allowed for us to increase the number of species that we actually have reference genomes for. And so if we see the relationship between the cost of sequencing, this is a little chart from The Economist in 2020, an article that was published there. You can see in blue we have the decrease in the cost of sequencing, and we have a few sort of technological advancements here. We've got pyro sequencing and single molecule real-time sequencing, nanopore sequencing, all this other stuff dropping the cost of sequencing. And as sequencing costs decrease, we see an increase in the total number of species that are actually being sequenced, whose genomes are being created. And we see that this is increasing rapidly into 2020, and it's just continuing to increase rapidly. There are now large genome sequencing projects, such as the Vertebrate Genome Project or the Earth Biogenome Project, where they're trying to sequence all 70-something thousand species of vertebrates, or all 1.5 million species of organisms that are on the earth, right? So big endeavors here, and the decrease in the cost of sequencing is really causing this field to explode. And the projections as we move forward, if we just think about humans, here we have something plotted on a log scale, but this was posted in 2016 about sort of, what was the targets for human genome sequencing, right? And for a long time, the goal has been able to get a human genome for just $1,000, right? And here they said, well, we think by 2020, we hope that we'll have about 50,000 human genomes if we just sort of increase linearly. But, if we go by how the historical rates are going, we expect to have something around 500,000. Well, just doing some sort of rough data amalgamation in public databases like Nomad, they've already been able to identify about 150,000 genomes sort of sequenced by 2020. So maybe we can get to 500. We might be. There's been a lot of genomes out there we might not know about. But within 10 years from now, just in the U.S., we're expected to have at least 1 million genomes just done by the NIH as part of the All of Us initiative. And that doesn't count everything else that's being done as part of medical science, as part of other research opportunities. And so, as we think about where the world of sequencing is going, and, we kind of got sidetracked by COVID, but there's going to be a huge continued increase in using genomic data within all fields of biological sciences. So this process is just going to keep on increasing, and this not only led to like a lot of increase in data, right, which was much easier to acquire, but it really led to a revolution in terms of the conceptual frameworks that we're able to think about, and big questions like what's a species, or what's an organism, or is the inheritance of acquired characters real? So are the things that we observe to be associated with evolution really actually being actualized and supported by the data? And these were really interesting things that we could now dig into in all kinds of ways that we hadn't been able to in the past, because the data was so much easier to acquire. And what this largely led to was the advent of a field called comparative genomics. Now, comparative genomics had been around for a while, right? This wasn't something that just started popping up in the early 2000s, but it really hit its stride then. So people have been working on chromosome level work for a while, had been doing comparative genomics, but it had been a lot tougher. Because in fact, before the field of comparative genomics had sort of come into vogue, we had spent a lot more time focusing on the gene. And if we think about this, what is a gene, right? A good way to think of a gene is a unit of heredity. And that unit of heredity is then transferred from parent to offspring. And it has some sort of characteristic that it then conveys off to that offspring. We can also take the concept of a gene and make it a little more distinct in what we're talking about. We can call it, a distinct sequence of nucleotides, right, that form a specific part of a chromosome, the order of which determines the order of monomers or peptides or nucleic acid molecule that a cell or virus may synthesize, right? So we're thinking about the part of a genome that is actually coding for something that has a sort of functional role. Conversely, when we think about what is a genome, right, it's not the same thing as a gene. A genome is the complete set of genetic material present in a cell or organism. In a simplified way, you might think of it as oh yeah, this is all the genes, but it's more than just the genes, right? So it's not just the genes. We're talking about the complete set of genetic material. When we talk about the field of genomics, genomics is the branch of molecular biology concerned with the structure, function, evolution, and mapping of genomes. This is in contrast to the branch of molecular biology that's concerned with genes, right? Or genetics. So genomics is taking a more broader view. When we look at genomics, we're looking not just at one thing, we're looking at all things at one time. We think about a very traditional field of biology, this idea of comparative biology. Comparative biology has been around for as long as biology has been around. And basically the idea is that we use different lineages, or a cross-lineage approach, a way to compare different organisms from different lineages. And we look at patterns of natural differences and similarities, so variation and disparity, right? Where do we see differences? And we use that to understand the phylogenetic and the mechanisms and patterns that drive the diversity of life. So, what are the origins of the evolution of different traits? What are the mechanisms that evolved? We use different species and we look at what's shared and what's divergent between them to drive this pattern. Well, comparative genomics is the combination of comparative biology and genomics, right? So here we have a field in which the complete genome sequences of different species are compared for regions of conservation and variation, right? So in this case, we're looking and we're comparing genome sequences, so actual nucleotide sequences, and we're seeing, okay, what's the same and what's different? And what that allows us to do is to understand what are the actual genetic changes and mechanisms that underlie the diversity of life that we see around us. So it's a very mechanistic, it's a very reductionist approach to trying to understand how we see these big differences in nature being echoed and generated in the genetics of these organisms. I like to think of this idea of why comparative biology matters. If we think about something that Goethe said, which is, he who does not know foreign languages does not know anything about his own. So it's only by looking outside of ourselves or looking at other organisms in comparison to another organism that we can really see what's important. If you're always looking at one thing, you'll never know why, what is similar or different from something else, and you might not understand why those similarities and differences matter in different contexts. So to truly understand it, it's really important to look outside of the system and do that comparison. Lesk, I think, has a nice way of summing this up too when we think about the field of comparative genomics, and this drives a lot of his discussion in the book, which is that one of the most important principles is that if evolution conserves something, in other words, it stays the same among multiple species, then that thing is probably essential, right? Because if it was to acquire mutation and lose function or change, that would very much change the organism, right? And it might no longer become viable. So again, it's this idea that we look for similarities and differences among species to identify, what's essential just for the organism to exist, and what might be important for the differences that underlie the different things, the different diversity that we see in nature. Which brings us to a really important concept, which is the connection between genotype and phenotype, right? So when I talk about a genotype, right, I'm talking about DNA sequence, basically. When I talk about a phenotype, I'm talking about a collection of observable traits other than your DNA sequence. And so what's the relationship between these two entities? Well, it's incredibly rare that phenotype is just equal to genotype. There are very few cases, there are some cases, but it's pretty rare that like a genotype will just generate, one-to-one phenotypes. In actuality, we end up with an equation that's a little more complex than that. It says, well, the phenotype, how things look, right, the observable traits that you see in organisms, is actually a function of the genetic material, the environment that organism grew up in, the developmental processes and influences that organism experienced, and perhaps some factors that act on top of the genome, some epigenetic factors. And also there could be interactions between these different factors. There could be genotype, genetic effects interacting with environmental effects. So there's a lot of things that could be going on here one of the terms that you may or may not be familiar with, life history, refers to the integration of total life experiences in which an organism develops. And so this is sometimes seen as that interaction between genotype and environment. So in other words, what you observe in an organism might be different depending upon if an organism plant grows up in a lot of sun, or it grows up in without very much water. Whatever is the differences between genotypes. But that is the differences you would see are expressed in different environments based upon the qualities of those environments. Epigenetics refers to heritable traits that cannot be explained by changes in the DNA sequence. In other words, they're things that happen on top of the DNA sequence. And so if we think about, what we think, how are we contrasting these two kinds of understanding of genotype explaining phenotype, and then genotype plus environment plus life history plus epigenetics. You could think of this case here where we could have very specific plans, right? Genotype is going to code for something. We're going to have these blueprint for what the phenotype should look like, when in fact, right, it's not as easily done as that, and it's more like something like an oboe concerto, right, where how it actually sounds, the music that you actually get out, will depend a lot upon who's playing the instrument, about their training, about the humidity of that day and how it affects the oboe, about the conductor, about any number of things, right? And there's all these other factors besides just the music that's on the page that's going to affect what the song sounds like. So when we think about how do we get to a phenotype, it's very rare that it's just the blueprint approach from genotype. It's actually a whole bunch of other stuff going on. I think one of the important things to understand as we move from what would probably be more of your experiences in thinking about genetics to thinking about genomes is that we need to think about what is a genome. And how does that differ than your conception of genetics or a gene? And so it's important that we think about how a genome is organized. And there's a bunch of different issues when it comes to genomes. And when we think about different types of organisms, their organization can often be very different as well. So if we think about something like these issues of packaging and replication, right? What we see here is that if we have something like a prokaryote and we were to stretch its DNA out all in a single line, we'd have about 1.6 millimeters of DNA in this genome. Whereas if we take a eukaryotic organism, on average, you're going to end up with a DNA stretched out into a single strand of about 1 meter in length, right? So there's some major differences in the size here, right? So at least an order of magnitude larger in size for eukaryotic cells versus prokaryotic cells, right? And what we actually have to see here is that DNA, that genome, right, is that genetic material is packaged very differently between the two. So if we think about a prokaryotic cell, there's no nucleus. If we think about a eukaryotic one, there is. Prokaryotic genomes are generally circular in nature, whereas eukaryotic genomes form chromosomes with the help of histones around which they wrap. And then there is there's lots of compartmentalization and cellular substructures in eukaryotic cells, and not so much in prokaryotic cells. And in turn, the replication part of prokaryotic cells and eukaryotic cells differences too, where eukaryotic cells have some very explicit replication and cell differentiation processes such as mitosis and meiosis, whereas prokaryotic cells are a little bit more free in the process that they undergo in fission. So thinking about the eukaryotic example, right, we all know about the nucleus and that the DNA is kept in the nucleus. And then, we might think that we would go from, we have our naked DNA, our double helix, right? But then this DNA is not just actually in a straight line. It's actually it tends to be wrapped around these entities that these histones that form these nucleosomes that are formed by histones. And then once it's wrapped around those nucleosomes, it starts stacking up into like these solenoidal features. And where it's wrapped around these nucleosomes, it forms something that we refer to as heterochromatin. And what happens is that heterochromatin starts to wrap around itself. It really compresses down and eventually condenses itself into something we refer to as chromatin, which in turn forms the chromosomes. And the chromosomes are these entities with these arms that you can see bound in the center here at the centromere. This tightly packed entity is one of the reasons why that meter of DNA is able to be packed into that nucleus. When things are open, when they're not as packed up here as they are here, we actually refer to that chromatin, this part of the chromosome, as euchromatin or open chromatin. It's different than heterochromatin. So, that's how that we see that, that's where all the genetic information is, but what is actually in that genome? What are the components that make it up, right? And so, we can go back to your basic biology and genetics and talk about, what is the central dogma and how it's associated with the genome. And often, a lot of people when they start off biology, this is how they imagine what a genome is, that basically we're going to follow this process where DNA is going to give us RNA and RNA is going to give us a protein, right? And so we have our coding sequence in our DNA. It's then transcribed and then translated into an amino acid sequence based upon a codon structure, a 3-base structure, right? And then that forms an amino acid peptide chain, which is then, because of the chemical properties of those amino acids, folds into a protein that has a 3-dimensional structure, and then that protein does stuff, right? And so that's like the classic way that we think about, how genetics works. And, if we think of something like a prokaryotic genome, it matches up pretty well with how we think a prokaryotic genome works, because about 90% of a prokaryotic genome is actually coding sequence, is actually these genes, right, that are coding for these proteins. So there's not a lot of wasted space. It's a very gene dense process. And there are other entities in this genome, things like operators and promoters and enhancers and silencers that are involved with turning on or regulating which genes should be active and in what context. But it's a fairly straightforward process in prokaryotes. In eukaryotes, it's a little bit different, because when you're looking at a eukaryotic genome, depending upon the size of the genome and the species you're looking at, it could be anywhere from 1 to 67% of your genome is actually protein coding. Only 1% of your genome could be protein coding sequence. What the heck is the rest of your genome doing, right? Like there's all this space that's not being used, and so there's a lot of other things in the genome at that point. And initially, we would have sort of been pretty dismissive about those other components of the genome. However, as comparative genomics has evolved, we've learned a lot more about these sort of non-traditional areas of the genome that we might have had less focus on in the past. But just getting back to the central dogma part of it, one of the big things that changes about eukaryotic genomes, right, is that instead of having, continuous coding reading sequences in these open reading frames, now we have an open reading frame which is interrupted with introns, right? We've got the red areas here which are associated with the exons, and when they are transcribed into mRNA, we see these introns cut out and form just a single mRNA strand that will then be sequenced. Again, we have sort of these regulatory factors, but in this case, instead of regulating sort of multiple genes at one time, a lot of these regulatory structures are spread out, so they may just target one gene at a time or a smaller number of genes at a time. But again, this is pretty close to what we're doing, but you can start to see that there'll be more space between genes. So intergenic regions, we now have these intragenic regions that are not protein coding. So it leaves the opportunity for a lot of other stuff to get into the genome and maybe act in a different way. So if we think of just what's in a genome, what are the components? Well, if we think about genes, if we think about just the human genome, right, there's only about 2 to 3% of the human genome which is coding for proteins. And we generally think of proteins as the things that are doing all this stuff, right? They're actually the ones that are doing the majority of the actions that we would think of. However, if we look at the percentage of the genome that's actually transcribed, that's made into an RNA, that comes in at about 80% of the genome. And a lot of this stuff ends up being something that we refer to as non-coding RNAs. And these non-coding RNAs can be involved in all sorts of regulatory function for controlling which genes are actually being translated into proteins or having action within this or function within the cell. In addition to that, there's a lot of the genome which is that can be made up of what are known as pseudogenes, which are basically degenerate genes that no longer can produce a viable polypeptide. And a lot of these genes have their origin. There's some cases over here that show the example of how these pseudogenes form. They can be formed either through a process where there's a gene duplication, and then you end up with a mutation in one copy of the gene where you might lose the promoting region, and so this thing doesn't ever really get expressed, or it starts to mutate and become non-functional. The other way is you can have cases where you might have a virus come in and insert itself into a gene region, sort of subverting the original sequence of it. Or you can have cases too where mRNA can sort of be processed and inserted back in the cell's DNA by accident. As I mentioned before, there's a lot of the genome which is actually transcribed, but we don't always know exactly what their function is. Some of the contents of these transcribed regions that have sort of unintended function that we're still learning a lot about are things like microRNAs. So what happens a lot with microRNAs is you'll have it expressed and it forms this sort of hairpin-like structure, this RNA structure, and then that structure is cleaved with something, say, like dicer, right? And then once that dicer is cleaves this section, right, these two microRNA duplexes right here will start to break apart. And when these things break apart, they then often, they're duplicated genes. They might come from duplicated genes, so there's another gene copy of it that they're pretty similar to. They can then start binding to these other genes, right? And where they do this, where they bind to these other mRNA products of, say, like the original gene, what they can do is they can actually facilitate the degradation of this mRNA, and thus limit the expression and the translation of that mRNA to actually be active. So this is one of the ways that we have some unique structures, and really microRNAs are something that we did not know a whole lot about until the advent of genomics. In addition to these gene or genetic components, I think it's really important that we spend some time thinking about and talking about repetitive elements. And we talk a little bit about repetitive elements in some of our other genome classes where we talk about genetics. But if you think about it, our genomes, the human genome, for instance, I told you is only 2 to 3% coding sequence. It's about 45 to 67% repetitive element, and that's a the numbers sort of change depending on what you define as a repetitive element, but that's a lot of our genome, right? And so for a long time, this stuff we referred to it as junk DNA because we didn't think it really had much point or purpose. And there are different types. There are moderately repetitive DNA things like or moderately repetitive DNA elements like dispersed gene families like the actins and the globins, or histone genes. There's stuff that we don't know what their function is, like lines, signs, and transposable elements. And then there's things that are just highly repetitive, just like long series of dinucleotide repeats, so AT, 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 AT. And a lot of times these repetitive elements, these satellite elements or telomeric structures, are associated with very specific parts of the chromosome, say being close to the centromere or being in the telomere or something like that. But as much as we thought that these repetitive elements aren't important, they are actually some of the most interesting parts of our genome, because in recent years, since we've been doing genomics, and even before that, we were able to delve in and really show just how interesting these other components are and how important they are. So, going back to the 1940s and later into the 60s, the work of Barbara McClintock, she actually was one of the people who discovered these transposable elements. She ended up winning a Nobel Prize for it. But it's this classic case of Indian maize where what you see here is you have kernels where some are dark and some are yellow, right? And the ones that are dark have something known as anthocyanins in them, and the ones that are yellow do not have anthocyanins. And why don't they have anthocyanins? So remember, this is an ear of corn, all from the same plant, right? So should be the same genome in each one of those kernels. Well, what actually happens is there's a certain kind of transposable element, certain kind of repetitive element called a transposable element, often referred to as a jumping gene, which can move around in the genome. And specifically in Barbara McClintock's case, she was looking at a set of genes known as DNA transposons. And these genes sort of work in a cut and paste mechanism where they cut out of one area and then insert themselves into another area. There are also retrotransposons. These are transposable elements that are often retroviral in origin. And basically what happens here is they're more of a copy and paste mechanism. So they'll be in the genome, there'll be an RNA intermediate made as a copy, and then that'll find somewhere else in the genome to insert itself. And as you can see here, huge phenotypic effects that happen from these repetitive elements that are in the genome. So they can have a big effect on what we actually see in nature. And basically what happens in this case in corn is that these DNA transposons, they move around, and when they insert themselves in specific spots, they can disrupt the coding sequence for the anthocyanins and basically turn it off. And so, depending upon where that DNA transposon ends up, will affect whether or not what color kernel we see in these ears of maize. In addition to transposable elements, there's a whole other suite of different dynamic components of genomes. There's lines and signs, ERVs and LTRs. LTRs are specifically, I think, an interesting class of repetitive element because they clearly have such strong relationship with viral elements. And so LTRs, we call them, that stands for long terminal repeat retrotransposons. They have a very identifiable structure where they have this target site repeat and this LTR region, this TGCA region that flanks a set of sort of predictable protein domains, has GAG and POL and some genes that we would expect to see there, maybe integrase or reverse transcriptase or something like that. And this easily identifies these things as generally having viral origins based upon what these genes are. And so that means that there's all these viruses that have infected our genome. So what? So there's all these transposable elements. Why are they important to us? If we think about, our gene structure in the central dogma, what we have is this case where, we have these open reading frames, and we've got this regulatory sequence region, right? Well, but it's only making up, a small portion of most eukaryotic genomes. It's really only like things like yeast, where it's like 67% of the genome. Humans, 2 to 3%. So we've got all these other repetitive elements in the genome. What are they actually doing? Well, things that transposable elements can do is they can affect those areas that are related to gene regulation. They can often cause a sequence to be broadcast or upregulated, somehow affecting the regulation of that gene or promoting it by moving a promoter in an area proximate to where that gene is located. They can alter the properties of genes by inserting themselves into certain parts of the gene. They are often seen as the engines of evolution, because if you want to have a lot of change happen in your genome, these transposable elements, because they're jumping around their genome and sometimes taking pieces of the genome with them when they jump, are interesting ways to sort of make big changes all at once. And they're also important in chromosomal rearrangements, recombination, which is another exciting way to generate variation in the genome, as well as epigenetic modification. Because of the fact of their different origin, a lot of these repetitive elements, because of viral origin, organisms are trying to knock them down. So, make it so they can't be expressed. But they often have some interesting relationships in that process as well. So, how does this relate to sort of the function of organisms? Well, if we think about how a virus, first off, just gets into a system like a eukaryotic genome, right? We can have cases where a virus can infect an organism, and then we have this interesting case, right, where the virus, once it infects an organism, will begin to make copies of itself, especially these retroviruses, things like coronavirus. They'll make copies of itself. They'll then make this viral RNA. And then what can happen is there's this process of endogenization where, when it makes this proviral DNA, it somehow, pieces of it might get integrated into the genome of an organism, right? So when this happens and this piece of this genome gets inserted or endogenized into the host genome, if that happens to end up in the germline, then you can end up passing on through vertical transmission this mutation to your offspring. And what's interesting here is we have this process by which humans are actually acquiring this viral DNA into their genome via a horizontal transmission pathway, and then passing it on to their young through a vertical transmission pathway. And this ENV is referring to an envelope protein, or the spike protein that a lot of people talk about. And this comes off, I think, in a really interesting case of a set of genes in mammalian systems that are known as the syncytin genes. And syncytin is a gene that has a transmembrane protein associated with it, and then it's got this envelope protein sort of tagged on to the end. And what's really interesting about this is if we think about like what an envelope protein does in a virus, right? That envelope protein is on the surface of the virus, and that allows this virus to find a receptor on that cell and then fuse with that cell. So that envelope protein is the thing that allows it to bind to that cell, and then once it's bound to it, it can then infect that cell. Well, it's the gene syncytin. What makes it really interesting is that basically you have a transmembrane part of the protein on one end, and then you have this envelope protein on the other end. But instead of being attached to a virus, now it's attached to another eukaryotic cell. And this allows this cell to take this envelope protein and co-opt its function and use it to fuse with another eukaryotic cell, forming this sort of multicellular syncytial layer that comes from this fusion. And so it ends up forming something called a syncytium. So why is this gene and this fusion of cells important? Well, if we think about something like the development of placenta, which is a key innovation for the evolution and origins of us. One of the interesting problems that had to be resolved was how does a mother actually carry an offspring, which is a foreign body inside of her own body, without her body rejecting that offspring, right? Because it's not 100% hers. It's 50% the father's genome. So it's going to look a little bit foreign to the mother. So how does she allow this organism to persist? Well, one of the things that happens is during placental development, the mother will form what's known as a syncytial trophoblastic layer. So between the mother and the fetus, they're going to form this syncytial trophoblastic layer. Right, and this syncytial trophoblast will then allow the mother and the offspring to sort of be in contact with each other, but not really be in direct contact with each other. And again, what forms this, right? What allows this layer to form is actually a viral gene. So a pretty cool way to co-opt a segment of your genome that you may normally not be wanting to use or use for something else, and make it part of your own genome. And if you think about how this might apply to things like evolution, we could look at, say, placental fishes and see something similar where they have these high maternal matrotrophy index values means that these are placental fishes versus sort of non-placental fishes. So here we have a bunch of cases where we see in these placental fishes that have higher levels of maternal investment in their offspring, but also have a sister taxa that has a lower level of maternal investment. We can see that the level of gene expression tracks very closely with this level of maternal investment, right? For a lot of these species, not all of them, but a lot of them, right? So if we look at this envelope protein in this one envelope protein, right? We see one pattern. We can look at another envelope protein and see a slightly different pattern. And what we can see is sort of a gradient associated with how placental an organism is and how much these two genes are expressed. And this was work that I actually did back at University of Connecticut with a student, Lauren Almonte, went on to be a physician's assistant. And what's really interesting about this case is that you can see that sometimes it's one envelope protein or one virus that seems to be the most active, and in another case it could be another virus, right? And that even for this virus, like we have a very placental species, Heterorandra formosa, So in that instance, like the FPKM values or the gene expression values are pretty low, right? So it may not be this envelope protein, which is really the one that's involved in sort of forming these fusion layers. It may be a different one because there's lots of envelope proteins in the genome. One of the students I worked with there, Eli Pascal, and I looked directly at some of these areas, and where you see purple is where we were able to identify where there was expression of these of one of these envelope proteins, and you can see that where there was expression is in the area where we would these are basically the placental interfaces of this organism, whereas there was no signal of the antisense marker for that. And then if we looked really closely at where that interface was associated with this belly sac, what we end up seeing is in this area, especially in the ovarian follicle part of this interface, this is actually looks to be very much a fused cell layer. So what we see in that case, right, is something similar to what we see in mammals, where we have maternal area here, we have the fetal area here, right, and they're separated by this fused syncytial layer. And so it looks like what we see in fish is similar to what we see in mammals. And additionally, when we looked for where these purple sections found these envelope coat proteins being expressed, they were all expressed with these syncytial layers, right? So we have a lot of evidence to suggest that these genes are also forming syncytial layers in these fishes that have placentas. And what's really neat to us is that if we look at all the different types of envelope coat proteins that we're able to find, what we find is that different organisms tend to have different lineages of these retroviral genes that they're expressing. So some of them have one lineage and another one will have another lineage that they're expressing. And so what's really cool about this to us is that when we think about, what's the point of having this repetitive element stuff in our genome? Well, one, through the co-opting process, right, you're able to sort of rapidly evolve a strategy like having a placenta, a new complex organ, to allow for very different life history and evolutionary pathway. And so it's a really interesting way to sort of evolve rapidly. But then the other really interesting thing about it is that it's different viral genes in different lineages. So this would indicate that it's not just one lineage, right? That it's not just an evolutionary artifact, that it's actually a broader strategy that can be applied in different organisms in order to solve this problem of how do we provide post-fertilization maternal provisioning, or feeding on, or how does the mother feed the young. So you can just see these retroviral genes, these repetitive elements, have had huge effect. I mean, they're basically helping to be responsible for the evolution of a complex organ, which is really wild. So they're not just junk. They actually may have a lot of really important purpose. Another area that's I think really important when we think about why we want to look at genomes is by thinking about variation in genomes. Specifically, we think about variation as being viewed as hierarchical. So we've got taxonomic level variation, we've got population level variation, we have variation that's within an individual. So there's all these different types of variation that we can have. So when we talk about the different kinds of variation, what are we actually talking about, right? Well, there's different types of variation. When we think about genetic variation. So it's really important that when we think about, we've talked so far that this perception of what is the same and what is different between different things in this comparative framework. Well, understanding, how we're going to quantify that is really important. So one of the ways that we talk about variation is variation in copy number. So how many copies does one organism have versus another? How many copies does one individual in a population have versus another? Another way that we describe variation is through variability in the nucleotide identity of something. So we actually look at the sequence and see where the sequences are different. Specifically in genomics, a lot of times we're looking at what are known as single nucleotide polymorphisms, or SNPs. And then the other area where we see variation is in the linkages among variants. And so a lot of times this can be, linkages can be subverted or different among individuals or taxon because of recombination or other things that have changed the co-inheritance or the co-expression of various types of genes and genomes. So what causes variation to exist among genomes, right? If we think about cases where we are looking at, say, like macaques in the Mediterranean, right? We see that if we look at a haplotype network, we see that there's a bunch of macaques here in Morocco that are all very closely related to each other. There's a bunch of other ones over here in Algiers and other parts of Algeria that look also to be very closely related to each other, very different from each other, right? Well, what accounts for this difference that we see between these two? What accounts for variation? Well, it looks like these are two pretty subdivided populations. So it could very well be that this group over here was isolated from this group, and they've just gone through various rounds of drift and mutation over time and diverged away from each other, right? But it's that population structure that allows for that to happen. Another important concept in talking about genomics is its intersection with developmental biology. And I think it's important here that we understand that when we talk about a genome is just the blueprint, right? Like, it's just the information, it's just the code. How that code is actually implemented can be very different and dynamic depending upon what an organism experiences. And so we often refer to this sort of experience that the genome has as its life history. So thinking about the different life stages it faces, the different environments it'll face, the different tissues that a genome might be expressed in, all will change sort of the dynamics of what is actually implemented from the genome. So, sometimes when we're thinking about genomics in the context of developmental biology, we're really trying to track the similarities and differences in the developmental process during evolution by tracking changes and similarities in the genome. Right, so by doing this comparative framework, we can see what are the static and what are the dynamic parts of the genome and how that might be related to the phenotypic diversity that we see. So how might things like body plan evolve? So if we think about the phylogeny, you can think about why it's important to consider these genetic things is because if we don't, we could do things that are traditional and based our phylogenies based upon morphologies, and we just kind of match things that look similar to each other. So, choanoflagellates look like choanocytes from Porifera, so they're closely related. Cnidaria and ctenophore look alike. We have all these different kinds of worms that are closely related to each other, right? So there's different ways to do that. But if I were to show you these, a picture of these three organisms, and I would ask you to say, well, which ones are related to each other, closely related to each other, you would see a jellyfish, you would see a sea anemone, you'd see this kind of worm-looking thing, and you would probably say, well, the top two. Well, this Budenbrachia, even despite the fact that it looks like a worm, it looks like a C. elegans or something like that, is actually nested within the Cnidaria, right? So this phenotype that the organism has is very different from the genotype. where the genotype would place it, right? It doesn't place it up with, all the worms in this area, different parts of the tree, right? It's actually way down here in Nidaria. In addition to giving us information on, the correct placement of organisms so we can do the right types of comparisons, there are other things that we can use comparative genomics for. So, one of the classic cases is in looking at Hox genes, which are genes that are really important in early development. In flies, they're generally expressed in a way in which things that are more towards the head and the upper torso are expressed in this pattern, and they're structured where they're expressed anterior to posterior, and they're sort of organized in this way as well, where we express the green ones here first and then sort of move down the process. Well, what's interesting is that most or lots of metazoans have these same sort of Hox clusters. And so we can see that, that same sort of cluster is actually conserved even all the way to us, to humans. And it's one of the ways that we've been able to use flies to study actually, human healthcare and human biology, is by looking at, how these patterns of how mutational patterns or changes in the genome might affect the structure and function of the proteins that are created or in this part of the genome. And it also reveals some things about our genome in comparison to history too. Like, we can see, look at the Hox cluster. We have one copy in, things like flies, but we've got 4 copies in humans. This indicates that there's been some sort of pattern of genome duplication at some point in our evolutionary history. And we can track back through other organisms to see, when did that occur? In addition to sort of these questions about, the genome and how we code it and how it's structured and its different components, it's important to associate it with other areas of inquiry. So we were sort of alluding to the fact that, there's this whole area of evolutionary genomics, but proteomics is another subfield which is really importantly tied to genomics. And, proteomics obviously is the study of proteins, and one of the reasons why it's such an associated field with genomics, of course, is that it's actually a DNA sequence, right, which is coding for the protein coding sequence. So when we talk about proteomics, right, we have a big emphasis on form and function, and it really is that amino acid sequence which is going to drive the creation of both secondary, tertiary, and quaternary structures. So understanding the relationship between proteomics and comparative genomics is really important, especially when we put it in the context of protein evolution, right? So where do we see differences between proteins? Generally what we're looking at are things that are defined either as insertions, deletions, or substitutions in sequences when we compare them to each other. So say we compare 2 sequences and we notice that there's been some stuff is similar between them and some stuff is changed, right? We'd be able to define those mutational changes either as an insertion, deletion, or an indel, or some type of substitution. If it's a coding sequence, one of the nice things that we often want to look for is whether or not that coding change, that change in sequence actually leads to a synonymous or non-synonymous mutation. So synonymous mutation would be a case here, right, where if I had a TTT and I changed to a C, right, it's still going to be a phenylalanine. And if I change from a TTT to a TTA in my sequence, right, I'm going to change to a leucine as the coded amino acid. So these types of changes can have a big effect on the form and function of a protein sequence. Another really important thing to consider and place where we're understanding sort of the order of genes and their relative placement to each other can be in understanding how patterns of duplication have occurred over time. So if we think about the case of various types of globin genes, in humans we've got both an alpha globin and a beta globin gene cluster. And how did we get, like at one point there was some sort of shared ancestor for these things, but at some point they diverged and they even end up on different chromosomes. And by looking at the placement of these genes and then comparing it to other organisms, we can start teasing apart, which one of them are actually the origins are of these suites of other genes. What is the pattern of duplication and divergence and evolution that we see among them? And what we can see here is we end up building something like a multiple sequence alignment. We compare the things that are similar and the same, and we line up the things that are different. And then based among this process, right, we can see that, that the human and the horse alpha hemoglobin, so these top two, right, tend to be more similar to each other than the way to the third and the fourth, which is the beta chain, right, where we saw there was a lot more things that were similar to them, but they still lined up, right? I mean, they still lined up with each other. They just weren't perfect. And one of the last areas that I'll talk about today is the concept of regulation. So we talked about the importance of protein sequence, as protein sequence are things actually doing things, DNA is coding things. Well, RNA is the thing which sort of bridges the gap. And regulation, so the process that either turns RNA off or turns RNA on, is thought to be incredibly important in how we actually control what proteins are being made. If we think about prokaryotes, they're traditionally regulated using that operonic system, that operon where they have this promoter region. If you remember from intro bio, right, you had like a rhabinose come in and bind up, and then it opened up that area, and then RNA polymerase would come in and start copying all these, transcribing all these genes and producing all that green fluorescent protein or whatever gene you want to put in that region. It's a slightly different process when we think about how this would be done. A lot more complex when we think about how it might be done in mammalian or eukaryotic systems. In this case, what happens is we would have these regions that need to be opened up in the same way that an operon opens up, right? But there needs to be some sort of modification under the chromosome to open up a region so that it can actually be got at. So there are modifications you can make to histones. There are other modifications you make to the conformation of the chromosome. All these things can affect whether or not the region that you're sort of targeting will be available for replication or transcription. An interesting case for like where regulation sort of ends up with very different things and how it matters is if we look at calico cats. So I don't know if you know this, but like calico cats are a good example for a concept that we know as X inactivation, where basically one X in mammalian systems is deactivated or rendered into a bar body, not to be used because of the fact that like basically when you have male and female systems and when you have XX and XY systems, there's going to be an imbalance in the number of sex chromosomes that males have. And so typically what happens is the female sex chromosome, one of the X's will become randomly inactivated in all cells. And this means that like the kind of like the gene dosage ends up being about the same, which is important. But one of the things that happens when you do this sort of stuff is you can end up with for these X-linked characters, you can end up with these genes or X chromosomes that have different characteristics depending upon whether or not they inherited from the mother or the father. And so when you look at a calico cat, like the orange parts and the black parts are typically means that, like, one parent was black and the other was orange, and now we mixed them together. And when we see this splotchy effect, what that is that is actually certain regions of the cat randomly becoming the father's genotype X chromosome, and other parts of the cat, the orange parts, being associated with the mother's part of the genome. And we refer to this process as genomic imprinting, where the parent of origin has a big effect on the actual phenotype. And what's interesting, though, is like if you look at the organism, if the kitten above that bigger one, what that actually is, that is his name is CC, or copycat. That's a clone of the cat on the bottom, but it doesn't look the same. So why doesn't it look the same? Well, that clone is actually made from a somatic cell, and they think that somatic cell already had the orange turned off on it. So even though the patterning is kind of similar, it doesn't really have the orange sample, so it won't be able to make the orange from say, like, it would have been inherited from the mother, so it can only make the black colors. Again, regulation could occur at lots of different checkpoints, and that's important to think about because it would affect how you might design a genomic study. But you could have regulation affecting transcription from DNA to RNA, from at the translation step from RNA to express protein, and there can even be regulation that can occur post-translation, where you need to have certain types of modifications, or there could be other types of inhibitions to the actual express protein doing its job. So it's important to know that there's all these different points where you could be looking at what is the role of expression. And when we think about what this regulation data can tell us, is we can start to pull it together and be able to identify groups of genes, groups of targets that are associated with different types of functions, different types of biological purposes. And so that's one of the things that I did quickly just to help us out. So that is the last slide of this lecture, of this first lecture, which is just meant to be a general background on some of the key concepts and background information that we're going to be talking about this semester. So I think it's really important that you just remember that you know what these concepts are and what they refer to, so that when they come up later, you'll have the appropriate context to be able to participate in the class. All right. Thanks for your time, everybody.